The human vascular system is a network of about 50,000 miles of tubes that carry blood. This blood continuously delivers oxygen and nutrients to the body's cells. The health of the vascular network is critical. Any vascular failure that shuts off blood to cells suffocates downstream cells, usually within moments. Vascular conditions that limit blood flow reduce the blood supply and trigger cellular distress and put cells into self-preservation. Factors that affect vascular health are important to the entire body. The vascular system is made of cells that also require nutrients and oxygen. Vascular cells experience blood like a bystander near an interstate and watch oxygen-rich blood cells zoom by. But blood velocity and CO2 swap mechanisms prevent vascular cells from accessing that oxygen bound to red blood cells. This means that vascular cells have to live on oxygen from someplace else. In fact, they must use the oxygen dissolved in the bulk fluid of blood, or plasma. Throughout medical history, plasma oxygen has been a curiosity. Medical texts recognize a normal concentration of about 3 milliliters per liter, tiny compared to the quantity bound to red blood cells. But tiny doesn't mean unimportant. This seemingly small quantity of oxygen keeps the vascular system alive. Until 1987, everybody except for Manfred von Ardenne overlooked the importance of this oxygen. Even now, it is not considered a factor in health or disease, even though nothing could be further from the truth. Plasma hypoxia is insufficient oxygen in blood plasma to maintain vascular cell metabolism. Conditions that reduce plasma oxygen below a critical level can damage the vascular system, just like the absence of oxygen damages any cell. One of the hidden effects of stress is respiratory compensation, which decreases the amount of oxygen dissolved in plasma. There are two known factors that govern usable oxygen available to vascular cells: flow and concentration. Flow is the volume of plasma that comes in contact with vascular cells, while concentration is the total amount of dissolved oxygen into the plasma. Increases in respiratory turbulence, like breathing hard, increase soluble oxygen levels, while shallow breath reduces levels. Dissolved solids, like salt, metabolic waste, reduce oxygen solubility in plasma. For example, seawater holds 25% less oxygen than freshwater, and blood plasma holds 43% less oxygen than even seawater. Conditions that increase plasma solutes or toxins decrease plasma oxygen levels. This is the hidden connection between blood toxins and the vascular system. Plasma toxins reduce soluble oxygen, and therefore reduce the vitality of vascular tissue. Blood is not water. It's a colloidal suspension governed by electrostatic repulsive forces. Suspended particles, like blood cells, must have enough electrical energy to repel each other. Otherwise, they form clumps that clog the vascular system. When clumps clog the narrowing vascular funnels, or arterioles, flow stops, and downstream tissue suffocates after the oxygen is used up. This causes cellular hypoxia, which transforms sludge blood into a destructive vascular inflammation. When the vascular inner skin or endothelium becomes oxygen deprived, energy metabolism failure disrupts the sodium-potassium ion exchange, and these cells accumulate sodium, which causes them to bloat. Bloating reduces the vascular diameter, which limits flow. The diameter of a healthy capillary is smaller than a healthy red blood cell, so that blood cells must bend or fold to go through capillaries. Bloating limits flow to plasma fluid and perhaps red blood cells that may shrink to avoid clogging the capillary. Chokes are a stuck effect because the oxygen required to release them cannot arrive because the pipe is choked. Manfred von Ardenne developed a system for releasing these chokes. We will talk more about that later. Logically, the lowest concentration of oxygen is at the very end of the pipe. In the vascular system, this is at the venous end of the capillaries, just before the blood starts its return journey to the heart. Ardenne and Leo discovered chokes exactly here, at the area of lowest oxygen concentration. The chokes were small enough to limit blood flow, but big enough to allow plasma to pass. This was the magic recipe for stuck. It was enough to keep the tissue alive, but not enough to thrive. Hence, stuck cell stress. Body cells fed by these capillaries enter a progressive process of distress after the capillaries choke. The reduced diameter forces the cells to survive on plasma, which still fits through the reduced diameter. The plasma carries glucose, so the cells switch to anaerobic or emergency power to survive. The cellular distress progresses as each body cell in the area has its energy decrease from 19 to 1, running without oxygen, so performance decreases by the same. These cells stop producing carbon dioxide, so the capillary valves that are opened by carbon dioxide remain shut. These anaerobic cells start to produce lots of acid, so the acids accumulate in the region and the area becomes permanently acidic. So the area remains permanently distressed in a low energy state, and this is the place where disease accumulates. These events create a local weakness anywhere and eventually everywhere in the body when they pile up. The role and location of each distressed area determines the effect. Cell weakness erodes performance, and acidic waste causes irritation and local pH imbalance. As more of the body enters distress, vitality decreases and health issues emerge. Distressed tissue in muscle is sore and weak. In the brain, causes motor and cognitive dysfunctions. In the immune system, increases infections and vulnerability, and so on. Large amounts of distressed tissue create body-wide effects that interfere with glucose, pH, and other regulatory systems, including the immune system. We're going to address these effects later. These distress effects from capillary chokes tend to be permanent because the metabolic circumstances that reverses them rarely occur. Remember the plasma oxygen we talked about earlier? Well, in the next section, we will explain how to use it to release tissue distress.